Well, I believe we finished our conversation last week with the discussion, completed discussion, I'm sorry, of the hormones, the adrenal cortex, which brings us to the hormones, the posterior pituitary. Now, I want to remind you something that we talked about before. Posterior pituitary does not produce hormones, right? It's a storage site for the hormones of hypothalamus that get delivered to the posterior pituitary and stored there. Essentially, here's the connection, and this is the time when we're going to compare posterior and anterior pituitary. So this is hypothalamus, and those cells that are shown here are essentially neurons. Those neurons extend their axons all the way into the posterior pituitary. So far, you with me? Axons essentially release hormones, two hormones, oxytocin and antidiuretic hormone. Okay? They release these hormones inside the tissue of posterior pituitary. In posterior pituitary, those hormones are picked up by the bloodstream and enter the general circulation. Does that make sense to you? So you see that here's the anatomical difference between the anterior and posterior pituitary. In case of anterior pituitary, hormones that are produced in the hypothalamus, they go into the anterior through the blood and stimulate other hormones in the anterior pituitary. Does that make sense? Like TRH goes to the anterior pituitary and stimulates TSH. In this case, hypothalamic hormones essentially are delivered into the posterior pituitary gland from which they enter the circulation. Clear? Now this pathway, these neurons are called hypothalama, hypophyseal tract. So here lies a difference between the delivery method to the anterior and posterior pituitary. Hypothalamo hypophyseal portal system delivers hormones to the anterior, so hypothalamo hypophyseal portal system, and hypothalamo hypophyseal tract delivers to the posterior. We clear? We're good? Now what are these two hormones? One is oxytocin, which essentially stimulates the process of childbirth, the actual delivery, the actual labor. Oxytocin is released from posterior pituitary. You see, I'm not saying it's synthesized, it is just released from the posterior pituitary in response to the pressure that baby's head exerts on the uterine cervix. When baby's head pushes on the cervix, the signal goes into the posterior pituitary and releases oxytocin. Oxytocin stimulates uterus to contract, increasing the pressure on the uterine cervix, which stimulates oxytocin, which stimulates uterus to contract. So this vicious cycle, what kind of feedback that is? Positive. Very good. So, and as any positive feedback regulation, it ends up with a pretty dramatic effect, which is baby gets delivered. That makes sense? So you have two people instead of one, essentially. Now, other roles of oxytocin aren't, well, except for related to babies, they aren't really well elucidated. So, oxytocin is necessary for breastfeeding. Before we mentioned the hormone produced by anterior pituitary called prolactin. I don't even mention what it does because it's so obvious, right? It stimulates the production of milk. But turns out that milk that is produced in the mammary glands cannot be released without the action of oxytocin. Oxytocin stimulates the contraction of the ducts in the alveoli in the mammary glands, and contraction of these ducts and alveoli pushes the milk toward the nipple. Does that make sense? So you see, you gotta have two hormones to enable breastfeeding. One that stimulates milk production, and another one that stimulates milk ejection. Clear? It is known that oxytocin increases in men during ejaculation. Is there any physiological importance to it? We have no idea. Okay. Now, antidiuretic hormone, or ADH. Let's talk about the name. ADH, antidiuretic. What does that mean, antidiuretic? Anti means prevents you from doing what? Peeing. Yeah, releasing water. It prevents you from peeing. Okay. Now, if this hormone, antidiuretic hormone, prevents you from peeing, what is essentially the function? What does it keep in the body? Water. Now, if it keeps water inside the body, what should be a stimulus for keeping water inside? Dehydration. When you get dehydrated, okay, your blood volume goes down. Does that make sense? Another thing is, when you get dehydrated, you lose water, right? Like with a sweat, for instance, okay? What happens when you lose just water? What happens to the concentration of solutes in your extracellular fluid? You dehydrated, you lost water. All your extracellular fluid, all like blood and stuff, they get more concentrated. We say that it's osmolality. The concentration of solutes goes up, okay? So essentially, stimuli for the release of ADH, either increased osmolality, or not necessarily and, or low blood volume and blood pressure. Does that make sense? Let's say it's increased osmolality, it's dehydration. When you get dehydrated, antidiuretic hormone is released, and it increases the retention of water in the kidneys, reabsorption of water in the kidneys. Water gets reabsorbed from what's going to be urine, right? When water gets reabsorbed, what happens to the volume of the urine? When water returns from what's going to be urine back to the body, what happens to the urine volume? Decreases. What happens to the urine concentration? Increases, right? Because smaller volume, same amount of solids. So when ADH is released, you produce very little urine that is very concentrated. Exactly what happens to you when you're dehydrated. Does it make sense? Another stimulus, which is decreased blood volume and blood pressure, leads to the same exact result. Usually your blood volume goes down when, say, you lose blood. Okay, like you donate blood or you get injured and there's bleeding. In response to this, antidiuretic hormone will increase water reabsorption in the kidneys. Okay? Your blood volume will go up, and your blood pressure, in response to this, will go up as well. Does that make sense to you? Now, alcohol inhibits the action of antidiuretic hormone on the kidneys. It's still produced, it just doesn't work. So, how does it change the volume of urine? If you drink alcohol and it inhibits actions of antidiuretic hormone, how does it change the volume of urine? It increases, yes, you pee more, and, the concentration of urine, yes, it goes down. It will be more diluted. Exactly. Does that make sense? So, it's one of the best, I think, one of the best examples, the best questions in endocrine physiology. So you tell somebody, okay, you have 2-3 beers, okay? You're going to produce a lot of urine just because of the sheer amount of fluid that you drink, right? But you have a few shots. You're going to produce a lot of urine because of inhibition of ADH. Does that make sense? So you're losing a lot of water, and this is what's responsible for the hangover. Dehydration that is due to the inhibition of ADH. Does that make sense? Now, there is a disease associated with a chronic underproduction of antidiuretic hormone. It's called diabetes insipidus, when the patient experiences insatiable thirst. He has to consume large volume of water. But it all comes out because ADH does not lead to reabsorption of water in the kidneys, so it's just basically cycling through. Does that make sense to you? Another effect of vasopressin or ADH is that it leads to constriction of blood vessels. And that makes perfect sense in light of what is the stimulus for ADH production. Look at this. One of the stimulus for ADH to be released is low blood pressure. Are you with me? In response to low blood pressure, ADH will increase water reabsorption, and it will also lead to vasoconstriction, constriction of blood vessels, right? When blood vessels get narrower, smaller in diameter, what happens to the blood pressure? Goes up. Does that make sense? So it's actually, in endocrine system, you should always look, okay, here's the stimulus. If we know the stimulus, we should know the effect. If we know the effect, we can figure out the stimulus. Make sense? Any questions about this too? Which brings us to a quite unique endocrine organ, the pancreas. The uniqueness of this organ is that it is both exocrine and endocrine gland. Pancreas does produce several hormones, but in terms of digestion, pancreas produces various enzymes indispensable for breaking down the food in the small intestine. We'll get to this when we get to digestive system. Now, let's talk about the hormones. Hormones are produced by endocrine cells located in so-called Langerhans islets. Those, flower-like shapes are essentially Langerhans islets. You with me? Those are the only endocrine structures in the pancreas. Langerhans islets, consist of 4 major cell types: alpha, beta, delta, and PP cells. Beta cells are the most numerous, then alpha cells, and then delta. I reserve the right to ask you about this 3. I'm not going to ask you about the function of PP cells in the exam. Okay? So, what are the functions of the enzymes? Glucagon. Oh, all of these enzymes, all of the pancreatic enzymes are water soluble. All of them. Okay. So glucagon, it produces pancreas produces glucagon in response to the low blood glucose. What is the effect then? Elevated blood glucose. It produces insulin in response to high blood glucose. What is the effect? lowers blood glucose, right? Now, somatostatin is produced in response to hormones like ghrelin and urocortin. Ghrelin, which we didn't talk about yet, but I can tell you. Ghrelin is the hormone of hunger. Okay? Hunger. When you're hungry, ghrelin is produced. Okay? And turns out, in this case, ghrelin is when you're hungry. Okay? Yes? Ghrelin? Here. Okay? So, when you're hungry, ghrelin is produced. It stimulates the release of somatostatin. Sorry, just give me a second. So, somatostatin is released, and somatostatin will inhibit insulin. When insulin gets inhibited, what happens to the blood glucose? It goes up. Does that make sense? So it can be maintained elevated when you're hungry. In some way, somatostatin, in this case, works in the same way as glucagon does. That makes sense? Inhibition of insulin will lead to elevated glucose. Good. Okay. Any questions about this? So, like, what is the product of beta cells? Insulin. Okay. What is the product of alpha cells? Glucagon. Questions like that. We good? So we're going to start with insulin, and we're going to talk about what it is, what's the signaling, and so on and so forth. Let's talk about insulin molecule first. So this is an actual insulin. This molecule is called proinsulin. So proinsulin gets partially cleaved by the enzymes, and that's the final protein. I used to have a table of all Nobel Prizes associated with insulin. So, Florian Chain. No, not Florian Chain. I forgot the names. I'm terrible with names, to be honest. But scientists that discovered insulin got Nobel Prize. Then the guy that determined the structure of the insulin, or guys, I don't remember, got Nobel Prize. Then the lady that developed radioimmunoassay to determine insulin got Nobel Prize. And then I believe the dude that synthesized insulin from scratch got Nobel Prize. So it's really Nobel Prize rich chemical. Okay. By the way, some of those studies for insulin discovery were done at Case Western Reserve University here. Now, what is the mechanism of insulin release? How do cells in the pancreas know when to release insulin? This is a pretty fascinating mechanism. It all starts with elevated blood glucose. Now, glucose is polar hydrophilic molecule. If it's polar and hydrophilic, can it cross the plasma membrane directly? Polar and hydrophilic. Can it cross the plasma membrane directly? No. Plasma membrane is hydrophobic inside, right? So it's got to have some kind of a transmembrane protein. You with me? Turns out there is a carrier called GLUT2. So glucose uses this carrier and enters beta cell. Now, when glucose in beta cell, its levels in the beta cell go up. Glucose is used by the cells as the molecule for ATP synthesis, right? Glucose is broken down in the cellular respiration, right? Which ultimately results in a molecule of ATP. before we move on. Is that clear? Is that clear? So more glucose, more cellular respiration, more ATP. We're good? And ATP, in this case, works as a ligand for a channel. There is an ATP-gated potassium channel. When ATP levels rise, ATP binds to the channel and closes it. Does that make sense so far? So glucose, let's recount it. Glucose gets into the cell, is used to make ATP. The more glucose, the more ATP. You with me so far? The more ATP, the more potassium channels are closed. Concentrations of potassium are higher on which side of the membrane? Intracellular. So if potassium channel is closed, potassium cannot leave the cell and stays inside the cell. Are we clear so far? Potassium is positively charged. So that positive charge leads to, leads to depolarization of the beta cell. Does that make sense? Depolarization of the beta cell, just like with the neuron, leads to the opening of voltage-gated calcium channels. We good? Concentration of calcium is higher on which side? Outer, right? So calcium diffuses into the beta cell, and in neurons, as you may remember, diffusion of calcium into the neuron led to the release of acetylcholine from vesicles. In the beta cells, diffusion of calcium leads to the release of insulin from the vesicles. So far, does that make sense? Now let's walk this thing through again. This time I want you to kind of say to me, speak out loud. So when glucose gets elevated in the extracellular fluid, it diffuses through the into the beta cell through the GLUT2, which is the carrier. When glucose goes up in the beta cell, how does it affect the levels of ATP? They go up. When levels of ATP go up, what happens to potassium channels? Why? Because these channels are gated by ATP, right? ATP is the signal for the channels to close. Does that make sense? When potassium channels close, what happens to the intracellular concentration of potassium? It increases. How does it change membrane potential? Cell depolarizes. How does depolarization affect calcium channels? Opens the voltage-gated calcium channels. What happens to calcium then? It gets into the beta cell and leads to release of what? Insulin. I find this pathway extremely elegant. Okay? So basically, all these multiple steps show you that elevated glucose leads to elevated insulin. Now, you have a lot of glucose in the blood. In order to lower it, what do you have to do? Where do you have to direct it from the blood? Any idea? You have a lot of glucose. You ate a cake. You have a lot of glucose in the blood. How to lower it? What does insulin do? Like which process does it stimulate? Where does glucose go from the blood? In the cells. Excellent. So glucose gets taken up by the cells. Does that make sense? Insulin essentially stimulates all the processes that lead to either redistribution of glucose into the cells or use of glucose. Let's take a look. So this is molecule of insulin right here. Okay. these blue ones. Okay? So these molecules of insulin, they bind to the insulin receptor. Insulin receptor works through tyrosine kinase. It's basically a part of the receptor right here. Okay? And tyrosine kinase, I'm sorry, phosphorylates a bunch of proteins inside the cell. So far, are you with me? And this phosphorylation leads to certain intracellular processes. First and foremost, uptake and storage of the glucose. Let's figure out what happens. So uptake. You understand the word uptake. Glucose gets into the cells of the liver and into the muscle cells. Good? What is the role of glucose in the cell? What does it do? What is it used for? You just mentioned it. Yes, ATP synthesis. So the first step of cellular respiration. Anyone? No? Well, the first step of the whole cell resp, there are 3 steps. The ATP is, no, in this case we're talking about like cellular respiration produces ATP actually. Okay? In cellular respiration, glucose is broken down, and the energy of that breakdown leads to ATP synthesis. The first step starts with G, glycolysis. So, the more glucose you have, the more glycolysis you have. Does that make sense? So that glucose uptake leads to elevated glycolysis levels. We clear? Now, if you have too much glucose, way more than you need, how do you store glucose? In the form of which molecule? Glycogen. So, glycogen, synthesis of glycogen is called glycogenesis. And if you still have, like, you filled your glycogen storage capacity to the top, how do you store even more energy? What do you convert all the excessive glucose into? Fat. So, lipogenesis. Does that make sense? Now, let's see. We've got insulin here bound to the receptor, and it stimulates, this binding stimulates phosphorylation of activation of proteins that are responsible for glucose uptake, glycolysis, glycogenesis, and lipogenesis. Does that make sense? Does it make sense? Now, let me ask you this. What is the opposite of synthesizing glycogen? Just tell me a phrase, like, what's the opposite of synthesis? Breakdown. So, if glycogen synthesis, glycogenesis is stimulated, then glycogenolysis, breakdown of glycogen, will be inhibited. Does that make sense to you? Now, glycolysis is the breakdown of glucose, right? What is the opposite of glucose breakdown? Huh? Yeah. So, the opposite of glucose breakdown is making glucose from scratch. This process is called gluconeogenesis, right? So, gluconeogenesis, making glucose from scratch, will also be inhibited. Do I make sense here? I will ask you questions. Something like, which of the following processes is stimulated by insulin? And I give you a list, like glycogenesis, or gluconeogenesis, or glycogenolysis, or lipogenesis. And you pick one or the ones that are stimulated by insulin. Am I clear? And I require to know the terminology. So during the exam, if you come to me and ask, what is gluconeogenesis? I will not tell you. Am I clear? I'm not trying to be mean, it's just something that you need to know. So, let's talk over it again. Just step by step. when glucose goes up, insulin goes up. What does insulin stimulate in the first place? Glucose from the blood, where does it go? Into the cells, right? Glucose uptake. It stimulates glucose uptake. Next, what does it stimulate in those cells where glucose is accumulating now? Glycolysis, the use of glucose. What else? Glycogenesis, the storage of glucose, and lipogenesis, even more storage. And it inhibits what? Glycogenolysis, breakdown of glycogen to make glucose, and gluconeogenesis, making glucose from scratch. You already have plenty. Clear? Does that make sense? Any questions about this? I hope I made myself really clear. So, some physiological effects. What stimulates insulin secretion? Blood glucose, blood lipids, blood amino acids. All of it, when all of it is elevated, insulin is released. More general, when does your glucose go up in the blood? After what? Yes. When you eat and food gets in your digestive tract. Even before it gets digested, your digestive tract produces a bunch of hormones associated with eating. Does that make sense? Like cholecystokinin or secretin. These hormones will stimulate the release of insulin. Does that make sense? So basically making you ready for taking the glucose from the blood into the muscle cells or liver cells. Am I clear? What inhibits insulin? Decreased blood glucose. Any kind of fasting. Okay. Anything that makes your blood glucose to go down. Does that make sense here? Now, there are a few kind of interesting graphs that I wanted to talk to. So this one shows you the levels of plasma insulin after some kind of a glucose challenge. Don't do it. Okay. So basically, if a patient drinks some sweet water, insulin spikes and then starts to accumulate over time to, stimulate glucose uptake. Does that make sense? Not only this, insulin levels are dose dependent. When levels of glucose in the plasma go beyond what's considered to be a physiological norm, which is about 100 milligram per deciliter, which is 100 mils, okay? Insulin levels start to rise dramatically. The more glucose is in the blood, the more insulin is produced. That makes sense? Why I say physiological norm? If you do a blood work, like you go to a physician, they tell you, go and give the blood, we're gonna do all the blood work. You have to do it in the morning. It should be fasting. Because if you do it after the meal, the glucose can be extremely high. That makes sense? Especially if you eat something really, really sweet. Now, interesting thing is, one of the most staggering evidence to the role of insulin in the glucose uptake is right here. So y-axis is intracellular glucose, glucose inside the muscle cell. Here, on the x-axis, is extracellular glucose, basically glucose in the blood. So this was done in the rats. If you take a rat and remove pancreas from it, okay, you can feed the rat with enormous amount of sugar, and rat muscle cells will not be able to take glucose in. Does that make sense? Can you see that? No matter how much sugar you give to the rat, muscle cells cannot take glucose because there's no insulin. If you look at the normal rat with insulin, or you supplement insulin to, rat after the removal of pancreas, Muscle cells can now easily take glucose in. Does that make sense? Not only this, turns out insulin stimulates uptake of amino acids as well. And that becomes crucial in athletes' diets. So this is the graph that was done not in athletes, it was done in the rats. So these rats got 2 glands removed. They got pancreas removed. And they got pituitary removed. So now they cannot produce growth hormone and insulin all at the same time. Are you following me? Now, if you would look at the dynamics of this rat growth, at the time when both these glands got removed, right here, the growth stunts. It becomes practically a flat line. That makes sense? Then, in this experiment, first, rats got injections of the growth hormone, which had very little effect on the growth. Then growth hormone was, terminated. Insulin started to get injected. Again, very little effect on the growth. Only when you supply rats with both growth hormone and insulin, rats start to become bigger, which makes perfect sense. Insulin stimulates the uptake of amino acids in the cell, and growth hormone stimulates the synthesis of proteins from these amino acids. Does that make sense to you? And in fact, in athletes' diets, if you would look at other bodybuilders, they carefully balance exercise and intake of carbohydrates. Sometimes they just shoot insulin, okay, to promote the uptake of amino acids from blood into the muscles to promote muscle growth. That makes sense? So insulin is a typical anabolic hormone. Any questions? Which brings us to a lovely story, which is diabetes. So, diabetes mellitus, I need to say, it's not, so diabetes insipidus is the disease associated with water balance problems. Make sense? Diabetes mellitus is the disease associated with the problems with sugar control. In diabetes mellitus, either insulin is not produced or insulin signaling doesn't work. Either way, if there's not a lot of insulin or insulin doesn't work, glucose cannot be taken into the cell. Does that make sense? So, you understand that? So basically the first physiological sign of diabetes, say in these depancreatized rats, elevated blood glucose levels. Can you see that over here? Because glucose is not taken into the cells. Here's another problem. When cells, like muscle cells or liver cells, do not receive enough glucose, can they use glucose for cell respiration if they don't have it? It's kind of a stupid question. No, they can't. So they got to use some other molecules, right? And those molecules will be fatty acids. So those liver cells, adipose cells, they will start to break down fat. Am I clear? One of the products of the fat breakdown, the most energy-bearing product of fat breakdown, are fatty acids. So immediately when insulin signaling or insulin production is impaired, you will see elevated levels of fatty acids in the blood. We clear? Now, when fatty acids are used as the energy source, one of the products of fatty acids break down are so-called keto acids, specifically acetoacetic acid. Another product of fatty acid breakdown is acetone. Have you heard an expression of acetone breath for people with diabetes? People with progressed diabetes that is not well controlled, they have very characteristic smell out of their mouth. They have acetone breath because they quite literally exhale acetone. Okay? That makes sense? But the concentration is so small that sometimes it is described as flowery. It has some kind of flowery note to it. Excellent. Yes. Another, by the way, we learned it the hard way. If your sick kid has a flowery breath, it is reason to be worried, but most likely it's dehydration. Okay? If they didn't eat long enough and they dehydrated, they're going to show signs similar to like diabetes. Okay? Just give them some water, give them some glucose, and they should be fine. Okay? If they're otherwise healthy. Now, one of the main, you can say, okay, whatever. What's the problem? Acetone, acetoacetic acid, who cares? Turns out that these compounds, acetoacetic acid and other ketones, lead to acidosis or diabetic ketoacidosis. Acidosis means that pH of the blood goes down. And how does acidosis affect the activity of neurons? It inhibits it. So constant lowering of the blood pH leads to inhibition of neuronal activity in the brain and eventually leads to coma. Of course, it doesn't mean that a patient who's diabetic misses an insulin shot, in 15 minutes this patient will slip into diabetic coma. But it's a very real risk for the patients with the diabetes. Does that make sense? Good? So understand the mechanism, right? Glucose doesn't get into the cells. Cells have to use fat. Products of fat breakdown are ketones. Ketones acidify the blood. Cool. Now, there are two types of diabetes. Type 1, type 2. Type 1 is also called a juvenile diabetes. It is usually early onset. So it's diagnosed early in life, like 4 or 5 years old. It's autoimmune. In juvenile diabetes, beta cells are gone. They're not there. Exact mechanism, why they are gone, we don't know. So no insulin is produced. Patients with type 1 diabetes have to inject insulin constantly. One of the main problems with that disease before was that, if a patient makes a slight mistake on the amount of insulin that he or she takes in, it may decrease the levels of glucose in the blood so precipitously that the brain will shut down because of not enough nutrients coming. Make sense? And in general, this fluctuations, if you think about it, like, you have really high glucose, take insulin, you have low blood glucose. Then you have high glucose, low blood glucose. This roller coaster is basically a biochemical stress. Okay, yes?
What happens if someone's glucose reading is up 300 or 400?
Mm-hmm.
What's the process? What's going on with their body?
We'll get to this.
Okay.
No. It's a great question. We'll get to this. I just want to finish the thought, and we'll get to this, okay? So this teeter-totter is not a good thing. And I actually read a quick, small article about the guy who is engineer, and I think his son has type 1 diabetes. And he was so tired of this syringes, he embarked on a adventure to make a device. Dyslexic. To make a device that would control insulin levels better. So basically, it's a pump, wearable pump, And insulin pumps are nothing new. You know, people wear them and they basically just press the button and insulin goes in the bloodstream. It's much more convenient. You don't have to actually inject, you know, manipulate with the syringes and stuff. But what he wanted to do, and he got a prototype. I don't know if he made it all the way to manufacturing. The device would control glucose levels in the blood and inject insulin when it is needed and as much as needed. Does that make sense? So if blood glucose goes up, it injects insulin. Just a little bit goes up, it injects just a little bit of insulin. It's a brilliant idea. I don't know if he actually made it yet. Okay. What happens if glucose levels in the blood are constantly up? Glucose in the blood gets oxidated. It's called, and glucose will also modify different proteins. That protein modification is named advanced glycation products, AGPs. AGPs stimulate inflammation in the blood vessels. Inflammation of the blood vessels leads to atherosclerosis. Inflammation of the retinal blood vessels leads to diabetic retinopathy. So it's basically a lot of vascular damage, really bad vascular damage. Okay. Now that's type 1. Controlled, you cannot do anything about it. Like, if somebody gets it, it's not their fault. Type 2 is late. And it's mostly associated with the lifestyle. I deliberately put genetics. I will focus on genetics a little bit later. But the mechanism is like this. If somebody constantly overdoes carbohydrates, say somebody has a breakfast, lunch, and dinner at the temple of diabetes. You know what it is? It's a cheesecake factory. So they have all the meals cheesecake factory and not from the healthy menu. Their blood glucose is skyrocketing every time. And that's the situation of the boy cried wolf. Constant overstimulation of insulin release and constant high insulin levels leads to the development of resistance right here. So insulin binds to the receptor, but signaling stops. Does that make sense? You know, like we all have this this callers, people, you know, companies that call us with some ridiculous stuff, and at some point you learn to recognize the number and you just stop picking it up. Okay, so that's basically what happens here. There's so much insulin signaling because of so much glucose in the blood that people develop insulin resistance. Am I clear? Now, when I say lifestyle, we all know what it means. It means Western diet, carbohydrate-rich diet. Okay, a lot of processed carbohydrates. So, bread, okay, sugar. That makes sense? Yeah, one of my rants, why I call Cheesecake Factory a temple of diabetes. I went there once. It was excellent, the greatest experience, one of the greatest experiences in my life. When you open the menu and you look, skip the fact that those slices of cheesecake bear minimum 1,400 calories per piece. When I got all the way through the slices menu and I got to the very last thing, sort of additions, it's not strawberries, it's not chocolate, it's not whipped cream. They have an offer. You can add 400 calories. I'm not kidding. Go to Cheesecake Factory and check the menu. You can just simply add 400 calories to a piece. They don't specify in what form, just 400 calories. Basically, if you want to die faster, go for it. Okay? Now, I have this word here, genetics. I mentioned before, probably a very good family friend, she's an endocrinologist. And we talk often about various, medical stuff. And she says, if you're genetically resistant, and there are people genetically resistant to type 2, you can eat all sugar in the world, like all sugar in the world, and you'll be fine. You will never develop type 2 diabetes. And there are some people who are more sensitive. So it takes much less for them to develop type 2. Does that make sense? Yes.
So kind of like just a little different. How could someone be like pre-diabetic? Because I know like people get diagnosed with pre-diabetic.
It's type 2. It refers to type 2. This is the situation when glucose levels are elevated. As far as I remember, clinically, it means that the fasting glucose is between 100 and 125. Which means they're not diabetic yet, but the pancreas does not control glucose levels as it should. Probably not because it doesn't produce enough insulin, but because the receptors, these receptors, already start developing resistance. And if you think about it, think about this. The person slowly develops type 2 diabetes. Fasting glucose levels go from 100 to say 130, 140. 125 and up, as far as I remember, is diabetes, okay? So say 140 fasting glucose. Now, this glucose, pancreas is functional, right? Pancreas works. So this elevated glucose, is it going to stimulate insulin production in the pancreas? Yes. So insulin levels, are they going to go up in the blood? Yes. But is there any signaling? No. So in patients with type 2, you're going to see elevated levels of glucose and elevated levels of insulin in the blood at the same time. Insulin is totally fine, yes.
What about gestational diabetes? When I was pregnant, they test you, they have you drink that nasty, like, orange fluid, and they test you for it. I ended up having like a 10-pound baby, so they were curious. They kept asking me, do you have gestational diabetes? I'm like, no. And they tested me 2 times for it, but they say due to having gestational diabetes, you're, I guess, more prone to having a bigger baby. But my glucose levels were really like healthy. So how does that work?
So basically, gestational diabetes, the mechanism for it is when you have a baby is a parasite, biologically, okay? It requires a lot of nutrients. And these nutrients are delivered by your blood to the placenta. And in order to supply baby with sufficient amount of glucose, not only amino acids, glucose as well, you have to break down a lot of your fat storage, or you have to eat a lot of sweets, okay? Consume carbohydrates in one or another form. All this sugar, it ends up in the baby, but before it ends up in the baby, it has to flow through your, systemic circulation, and it will stimulate your pancreas to release insulin, okay? So that actually is gestational diabetes, and it makes sense that it can develop if you carry a large baby, because large babies will require more nutrients to grow. Make sense? Remember we talked about cortisol that elevates blood glucose. Prolonged stress can lead to diabetes, type 2. Make sense? Now, I used to study obesity, and these 2 graphs, I took them from somewhere. My bad, I think it's something like Obesity Watch website, something like that. It's actually like really, really reliable data source. So they show you, this image on top shows you the distribution of diabetes percentage in the United States based on the states. Okay, and this is obesity. So the breakdown is always different, and it's sad to look. I'm gonna talk about obesity first a little bit. It's kind of sad to look at it because they have to introduce new categories. Up until a certain year, the highest percentage was 30. Now they have to introduce 35. But I did a lot of digging into obesity topic when I was doing research, and I can tell you, I call it an obesity belt. All southern states, from Texas through Louisiana all the way to Florida, Okay, that makes sense? Florida is a little bit better because there are a lot of Yankees, retired Yankees, that do not bring a lot of fat there. Don't take it as an offense. It's a sarcasm. And then you go here all the way to West Virginia, and you will see a huge proportion of obese patients live in Michigan. There are two sort of common themes that you can see associated with obesity. One is poverty. You know, Louisiana, Mississippi, Alabama, Georgia. Georgia is basically Alabama with Atlanta. So there's not much difference between those states. Poverty. West Virginia is a fairly poor state. Again, poverty. You may ask, why Michigan? Biggest city is Detroit. And Detroit is a large African-American population that is unfortunately fairly poor. And that is associated with that, basically like one city, but it gives a lot of percentage to the entire state. And on top of that, there's some ethnic differences. So people of the Pacific descent, like Hawaiians and Pacific Islanders, people of Mexican and generally, well, more Mexican Latino descent, Native Americans and African Americans are more prone to obesity. Interesting question. Why? Probably because they had lighter diets than Caucasians who are Northerners. As Northerners, they had to eat more, okay, and they less prone to developing, I don't know. So that's some ethnic discrepancy. But if you would look at the percentage of obesity, percentage of diabetes, you will see a massive correlation. That makes sense? So well-controlled body weight improves the conditions of people with type 2 diabetes. And that's what I want to show here. So this is called glucose tolerance test. The red line represents a patient with the normal glucose control capacities. This moment, patients either takes the injection of glucose or just drinks a whole bolus of glucose. And then patients' glucose levels in the blood are measured usually 15 minutes interval, bless you, 15 minutes intervals, and you can build a curve. That's one of the physiological experiments that you can technically do yourself. You can buy a glucometer, and you can make a solution, you can drink it, you can measure you have to start, it should be fasting, okay? Minimum 6 hours fasting before. And if you have normal glucose control capacities, okay, you will have elevated blood glucose, of course, then insulin kicks in and your blood glucose goes down. Good? Make sense? Now, you can see that it goes back to normal in about 2 hours. That's actually a pretty valid clinical parameter. Does that make sense? Like if in 2 hours your blood glucose is back to normal, you're all good. Plus minus 15 minutes. Patients with diabetes, first of all, fasting glucose is elevated. And glucose levels in the blood, they decrease really, really slowly. Much slower than in patients without. That makes sense? Now, if patient has a type 1 diabetes, there's, not much you can do. You can change your lifestyle all you want. That doesn't bring back the functional pancreas. In patients with type 2, weight loss and exercise. So this shows you the efficiency of 2 exercise regimens in controlling blood glucose and increasing insulin sensitivity. So let's clarify this term first. Remember I told you about insulin resistance, right? So insulin sensitivity is kind of an opposite. People who have good insulin sensitivity are not resistant to insulin. Insulin works. Make sense? higher insulin sensitivity is good, basically. So these are diabetic patients, and this is some kind of a score, of an index of insulin sensitivity. They start fairly low, and then they do exercises. Okay, they do a bout of high intensity versus low intensity exercise. High intensity are squares, low intensity are diamonds. And you can see that after a bout of certain time of high intensity exercise, insulin sensitivity increases, and then it basically goes back. Right? Interestingly enough, they do longer bout. That's important. Longer but low intensity exercise. Insulin sensitivity stays up for a longer period of time. What does that mean for you? It means that if you have a job that is, say, sitting all the time, to control your blood glucose, it is more beneficial for you to stand up and walk around the office or whatever for 1-2 minutes every hour than hit a gym once or twice a week for 30-35 minutes. I'm not talking about like strength or anything. I'm talking strictly about glucose control. Question? No. Say it again.
What causes sclerosis in someone who's not overweight?
By sclerosis, you mean atherosclerosis, vascular?
Yeah.
That's kind of normal. Like, we all have it. It's, oh, you mean abnormal atherosclerosis. In someone who's not overweight. Usually, it's genetic deficiency that, people have extremely abnormally high levels of fatty acids in the blood, or hypercholesterolemia. I have a very good friend who is a twig, okay, and he has to take Lipitor because his cholesterol levels are skyrocketing all the time. He has a genetic problem, His system does not control cholesterol as it should. Make sense? Did I answer? Good. Well, you can make it happen if you want. You have to switch basically to eating butter. And only butter. And a lot of butter. Okay? Then you have higher risk for developing atherosclerosis. But it requires a lot of dedication to do it by a diet. Usually it's obesity or genetics. Now, what about body weight control? Here, you have two graphs. This is the insulin levels, and those are glucose levels. That's a study in women, overweight women. They were placed on the caloric restriction diet, basically limited calorie intake, lost some weight, and that led to lower glucose levels and lower insulin levels, which are both good signs in type 2 diabetes. Does that make sense? And in fact, I know a few people, you probably know too, with type 2 diabetes. If people, take control of diets, they start to exercise, they don't really have to use insulin anymore. It can be controlled. Okay. Does that make sense? Now, we're going to talk about glucagon and then take a break. So, glucagon is the opposite of insulin, okay, in terms of the function. It's a protein. Here you can see glucagon binds to its receptor, and it stimulates a bunch of intracellular events. I'm not going to ask you, whether it works. It works through phospholipase C, adenylate cyclase. All you need to understand is that it's water soluble, so the receptor is on the surface. Now, glucagon will, increase blood glucose. Here you can see the lower the blood glucose, the higher the glucagon levels. Here in this graph in the upper left corner. Okay? The lower the glucose, the higher the levels of glucagon. Make sense? Now, think about this. If insulin stimulates glycolysis, glycogenesis, and lipogenesis. Then glucagon will stimulate what? In order to increase blood glucose. What should we do with glycogen that's already been synthesized? Glycogen is made from glucose in order to store it. Now we have a situation when blood glucose is really low. What do you have to do with glycogen? Break it down. We made glycogen in the times of abundance. Now we had a time of need, so we need to break down glycogen. So glycogenolysis is stimulated. In the time of abundance, we used all the glucose. What do we need to do now? Make more gluconeogenesis, synthesizing glucose from scratch. In the time of abundance, we stored glucose in the form of fat. Now we need to do what with this fat? If we stored it, now we need to break it down. So lipolysis. You see, processes that are stimulated by insulin are inhibited by glucagon and vice versa. Processes that are stimulated by glucagon are inhibited by insulin. Does that make sense? So basically, if you think about it, like how can we apply it to day-to-day life? If you will go on the really, really long run, first you're going to start breaking down glycogen that is stored in your muscle cells and liver. And once you're done breaking it down, what are you going to switch to? What are you going to break down? Fat. Yep, you're going to start breaking down fat. And if you're not used to it, if you did endurance running, you know what I'm talking about, not used to it, you're going to hit the wall. So that's the moment when your cellular metabolism has to switch from, using glycogen as the source of glucose to using lipids as the ultimate source of ATP and, make glucose from scratch. And it's hard. And we'll talk about it when we talk about metabolic processes and digestive system, okay? Does that make sense? I don't know how stress it more, but you're going to get questions. Which of the following processes, insulin or glucagon, stimulate or inhibit? And I'm going to use these fancy words like gluconeogenesis or glycolysis or lipogenesis, and you have to be absolutely familiar with those terms. Am I clear? Any questions? No? All good? Let's take a break.